Okay, welcome to Unit 3, which is covering Chapter 8, and we are dividing Chapter 8 into two videos. So Part 1 will cover the pages listed here. So Chapter 8 is all about the metabolism of microbes. We're going to start with a little primer on how chemical reactions are written, because we'll be dealing with a lot of them. In case you're not familiar with chemical reactions because you don't have a chemistry background, we'll just kind of review on this slide. So in a chemical reaction, we have reactants which are formed into products. So the molecules that are reactants are converted. So we have the arrow which indicates the conversion from reactant to product. And multiple reactants can combine together to form one or multiple products. The letters represent the atoms that we're dealing with in this particular chemical reaction. So here we're combining hydrogen and oxygen as our reactants to make water. The subscripts and the coefficients are to balance the equation, and we're not going to deal with that in our class. I do want to point out that every chemical reaction is driven by a specific enzyme, and that some chemical reactions are reversible, meaning reactants can form products or products can form back into reactants. And when you see chemical reactions that are reversible, you'll have a double-sided arrow going in both directions. We may also see molecules written in this form in some questions on quizzes or exams or in the chapter book. So this is a molecule of water with 2 hydrogen atoms and 1 oxygen. The lines in between them are just demonstrating that the oxygen and hydrogens are bound together, and it's written in this formula to show a little bit about the shape of the molecule. So metabolism overall refers to all the chemical reactions in the cell. These chemical reactions are either catabolic, meaning we take a large molecule and break it down, or anabolic, meaning we build a large molecule from smaller precursors. So catabolic reactions will result in the release of energy. And in Chapter 8, we're going to be starting with a carbohydrate called glucose, and we're going to discuss how the cell takes this large glucose molecule and breaks it down through a series of catabolic reactions into smaller molecules. And as those catabolic reactions are done, we're going to have a release of energy. And so anabolic reactions are building larger molecules, and those can be things like proteins, so we can make amino acids, and then those amino acids can be built into proteins. We can make nucleotides, which are the building blocks of DNA or RNA, so those are formed into those large nucleic acid molecules. We can build in the cell smaller fatty acids in an anabolic reaction and then combine those fatty acids together into another anabolic reaction to form complex lipids. And then all of these large molecules are then brought together and organized into an overall cell. So the building of all of these things requires energy. And so in this figure, we have the catabolic reaction yielding energy and the anabolic reactions using the energy that we got from the catabolic one. The linker between catabolic and anabolic reactions is often ATP, which is referred to as the money currency of the cell. It's how the cell stores and uses energy. So again, the catabolic reactions are starting with a large or a macromolecule and breaking it down into a simpler one. And this results in the release of energy. The anabolic reactions, on the other hand, are taking simpler molecules, having an input of energy, and then we build a larger molecule in the anabolic. So this energy released in a catabolic reaction is used to make ATP, and that ATP will store the energy until it's needed in an anabolic reaction, at which point the ATP is broken down. The energy extracted from ATP will then be used to build a larger macromolecule in an anabolic reaction. And so this is what we're doing in Chapter 8. We're focusing in on how the cell starts with food, a food source, and we're going to use the food source glucose because it's a very common carbohydrate that a lot of microbes use as a starting point for metabolism. We're going to talk about how glucose is broken down in a series of catabolic reactions, and each of those catabolic reactions results in the release of energy. That energy release will then be used by the microbe to build ATP, which can then be used in anabolic reactions or performing cellular work. So ATP, if we look at the structure of this molecule, is comprised of an adenine, which is the same nucleotide that is going to be present in DNA and RNA. It's made of a sugar called ribose, and then attached to the ribose are 3 phosphate groups. So that's the P surrounded by oxygens and hydrogens. The 3 phosphate groups have a negative charge, and those negative charges are held together side by side, which is a significant amount of energy to hold 3 negative charges that would like to repel together in the same molecule. So ATP has a significant amount of energy because of these 3 phosphate groups. When we want to get the energy out of ATP, what the cell will do is remove the 3rd phosphate. So then we're left with 2 of the 3 phosphates, and the molecule will be called adenosine diphosphate, or ADP. And when that 3rd phosphate is broken off, energy is released, and that energy will be used in anabolic reactions to build larger molecules. An additional type of reaction we're going to discuss in Chapter 8 is the redox reaction or oxidation reduction reaction. So these types of reactions involve the transfer of energy from one molecule to another. So this is a simple example showing the sodium atom as a reactant combined with the chlorine atom as the second reactant. And then the arrow indicates that those combine to form the product, which is sodium chloride. And so the reason this is a redox reaction is because the electron, which is illustrated by a gray circle, starting on the sodium reactant atom, will be transferred to the chlorine atom in this redox reaction. So redox reactions involve the transfer of energy in the form of electrons from one molecule to another. So here's another example of a redox reaction, and this is the redox reaction that we're focusing on in Chapter 8. So shown in this reaction are C6H12O6, which is the chemical formula for glucose, reacting with 6 molecules of oxygen to form 6 molecules of carbon dioxide and 6 molecules of water. So this is a redox reaction because in this reaction, the glucose is being oxidized to carbon dioxide and the oxygen is being reduced to water. So there's always an oxidation and a reduction coupled together in a redox reaction. So what's happening is the glucose molecule is transferring electrons and losing electrons to form carbon dioxide as a product. The oxygen molecule is gaining those electrons that were passed from glucose to form water as a product. So tracking electrons is something chemists are very good at doing. In biology, we're going to track the electron movement by watching where hydrogens go. So in this equation, we have glucose with 12 hydrogens, which will be converted in product form to carbon dioxide, which has no hydrogens. The electrons and hydrogens move together, so going from glucose to carbon dioxide, we have a loss of hydrogens. So the glucose is being oxidized, which means it's losing electrons or hydrogens to form CO2. The other half of this is the reduction. So here we have oxygen, which has no hydrogens as a reactant, forming water as the product, which has 2 hydrogens per molecule. So this represents a gain in electrons, a gain in energy, a gain in hydrogens. So oxygen is reduced to water in this redox reaction. And there's an acronym that can help you remember this. Oxidation is loss, spells oil, and oxidation is loss, meaning oxidation is loss of energy, loss of electrons, loss of hydrogen. The second acronym is reduction is gain, or RIG. So reduction is gain, meaning molecules that are reduced will be gaining energy, they'll be gaining electrons, and they'll be gaining hydrogens. So oxidation is loss, reduction is gain. Your acronym is oil rig. So I mentioned earlier that enzymes are driving chemical reactions, and each chemical reaction has a specific enzyme. So this role of enzymes is described as being catalysts. So let's define these two vocabulary words. Enzyme is a protein molecule that serves as a catalyst, and it functions for one substrate in particular. So this is why each enzyme is specific for each chemical reaction. Catalyst is a substance that speeds up a chemical reaction without being altered itself. So enzymes are proteins in the microbe in the cell that speed up chemical reactions, and when they do this job, they are not themselves altered, so they can be recycled by the cell and reused over and over. So if we look at the interaction between an enzyme and its substrate, we can go to figure 8.4 in the textbook. So in this figure, we have the enzyme as a purple blob. The gap in the enzyme, where there's kind of a hole, is the active site. And the active site of the enzyme is going to have a shape that's specific for the enzyme's substrate. So this substrate is really the reactant in the chemical reaction. It's going to be formed into product. So the words substrate and reactant are synonymous. They mean the same thing. So this particular enzyme has an active site that fits this particular substrate, which is labeled S. You can see there's another substrate that looks like a shark's tooth, and that substrate will not fit into the enzyme active site. So this enzyme is specific for this one substrate, which is going to be formed into some specific product. So when the substrate and enzyme come into contact, the substrate binds to the enzyme at the active site, and the enzyme will then help the substrate to form into products, which in this case means the substrate is broken up into two molecules. When the products are formed, they will be released from the active site, and then the enzyme can go and bind another identical substrate and catalyze that substrate to be formed into products. Enzymes are affected by temperature and pH because they have to maintain a very specific 3D shape in order to bind to their substrate specifically. When the temperature or pH of the cell's environment changes, it can cause denaturation or an unfolding of the enzyme. So if we have an enzyme that should look as it does on the left in this ribbon diagram, which is a way of showing 3D shape of proteins, and we alter the pH or temperature at which the enzyme is existing, the enzyme can denature and unfold, and the shape of the active site will be lost, so it can no longer interact with its substrate. There are ways to inhibit the function of enzymes, and these can be molecules that either competitively inhibit enzyme function or non-competitively inhibit enzyme function. So I'm going to explain competitive inhibition first. So we have the enzyme drawn in purple, we have the substrate in dark brown, and we have the competitive inhibitor in light brown. So the shape of the substrate and the competitive inhibitor are very similar, so they can both bind to the enzyme active site. When the substrate binds to the enzyme active site, the reaction proceeds and the substrate is formed into products. When the competitive inhibitor binds at the active site, it blocks the substrate from binding there and the reaction is blocked, so products are not formed. So both the substrate and the competitive inhibitor bind at the active site. In non-competitive inhibition, shown on the right in this figure, we have the enzyme drawn in pink and purple and the substrate in dark brown. We have an active site where the substrate will bind. So if the substrate binds to the active site, as expected, the reaction proceeds and product is formed. If we have a non-competitive inhibitor present, the non-competitive inhibitor can bind at a site away from the active site called the regulatory site. When the non-competitive inhibitor binds at the regulatory site, the active site changes shape and the substrate can't bind. Then the reaction is blocked and product is not formed. In this example, the product of the reaction is the non-competitive inhibitor. So if a lot of product is formed in this particular reaction, the cell may not require any more product, so wants to shut down the reaction. So the product will bind at the regulatory site, thereby altering the active site of the enzyme, and the substrate won't bind and the reaction is blocked.